That's absolutely fine. I'm presuming that everyone can see my slides, so I'm going to do a good afternoon and good morning. So for me, we're heading towards the evening, but I understand for some people we're only just beginning. My name's Charlotte, and I head up what is called the Oxford Protein Informatics Group, which is based in the Statistics Department at the University of Oxford. And one of our main research areas is around immunoinformatics. And I think the first thing I should say, given the community I'm speaking to today, and the pictures on the bottom of this title slide are meant to kind of remind me to say this, is that we as a group really started from this perspective of thinking about protein structures, and in this case antibody structures, nanobody structures, TCR structures, and have moved from that towards thinking about sequences. So I will show you on many occasions how we're trying to move from just having a sequence understanding of this, and I want to be careful, just isn't a great word there, but to painting on these levels of structure and understanding that might be really helpful going forwards. I'm going to start by mentioning a tool or a database which I hope at least some of you are familiar with, which is our observed antibody space. This contains now a large number of studies across diverse immune states, organisms, and individuals. And the big thing for us was to take the data so that we could look at it from this structural point of view. So to sort it, clean it, and annotate it, and in particular, translate it and number it, because in that state, you can then move on to doing lots of other types of analysis. And it preserves all of the fine-grained metadata. I think the thing I'm going to show you on the next slide is we've just expanded to try and capture data from 10x and other paired sequencing technologies, which is quite an exciting step for us. As I hope every speaker is going to say during these talks, this data is all completely downloadable. You can download the entire lot, or you can look for things that you're interested in and download those pieces as you see fit. So this is a very, very recent update that happened either late last night or early this morning, that we've actually managed to get some of the paired data into this format as well, where it's sequenced and numbered. And this is the set of studies so far that we've got in, and just showing you the kinds of species that's across and the data sets that we're moving to include within this. The next thing I'm going to talk about is something that we started doing, I think, towards the end of March was really when we began. And for us, it was a kind of vital step that was missing when we were trying to analyze data that might start coming out around the coronavirus outbreak, and also how we could use data that was coming from sequencing studies. The question was, how much did we actually know about coronavirus antibodies? So there are a huge number of preprints now on SARS-CoV-2. So about 1,000 had come out between January and March, another sort of 3,500 April to May. I haven't got the data for the most recent, but would expect that kind of increase again. And there's quite a lot of work on previous coronaviruses like SARS-CoV-1 back from 2003 and MERS in 2012. So we went out and searched the scientific literature for antibodies that had been stated to bind to one of these three, CoV-1, CoV-2, or MERS. And we looked across lots of different places, so not just publications, but also things like patents, because that could create quite a lot of information here. The sequence information for these was scattered all across the place, which made this quite a hard place to come and find this. And we were very thankful. There's a few of the reviews that are mentioned here to people who had attempted to do parts of this before. And we did this to generate this database where all of this was contained. And we're still working on keeping this continuously updated. So what does it contain? Well, roughly speaking, right now, there's about 1,500 entries in the database. And about 1,000 of those are linked to SARS-CoV-2. Now, obviously, those of you who can add up quickly will see that adds up to more than 1,438. But of course, some entries are linked to more than one of these coronaviruses. We're trying to collect the full FV variable domain sequences. We've got that for about 90%, and we keep trying to trace the rest. And there are some where it's marked where, in order to get hold of them, you will have to communicate directly with authors if you want them. The other very exciting thing for us is quite a large number of these, thanks to efforts in all sorts of places across the world. We've also got solved structures, so we know the three-dimensional structures of these antibodies as well, which allows us to talk about them in different ways. I'm now, instead of going to show you a dry set of screenshots or text, just do a little video of what's in the database so you can get an idea of what I'm talking about. Oops, that went well. I'm just going to go back. Sorry about that. Try and press play. Here we go. So this is just the front page of the database. It's fairly obvious what we're looking at. First thing to stress is everything is downloadable, the whole lot or particular data sets you're interested in or models. Mostly what I think people will be doing is searching by attributes you might be interested in. So here I'm going to search for antibodies that bind to SARS-CoV-2 but not CoV-1 and are neutralizing against CoV-2. I perform that search, I get 165 results back, so you can see them across here, and we've got the attributes of these things and also their sequences. You can see the VH and the VL, and we go right across to where we have their structures. In this case it's a homology model. The sources and things are across to the right there, and this is just a quick display of the kind of model. So in this case it's a model that we've built of that particular hit, but it could be a real known structure as well. The other thing you can do is search by sequence if you've got something you're particularly interested in. I'm just going to load our example sequence because that's a fast way of doing it, and it'll show you the close sequence hits to that sequence within the database, and you can see what they were effective, you know, what types of things that they were found neutralizing or not for. It also does the matches directly to the CDARH3 as well. So I'm hoping that's given you a flavor of the kinds of things it's possible to do with the database. I'm going to show you a very small number of results as well, just to give you an idea of the kind of information you can get out of this, even without comparing it to other data sets. So things you can look at are developmental origins. And one of the things here that was interesting to us is that over time, the origins are changing quite a lot, which means we're going to have to be very careful when we compare between different viruses, as people start saying specific behaviors related to, say, SARS-CoV-2 in terms of what types of things will bind. Well, actually, we haven't been using the same systems to look at them, so we may not have the same answers here. So we're moving towards a much higher percentage of antibodies being baited directly from human serum. So we're skipping things like the phage display stage or immunized animal antibodies, and we're seeing much more of this baited directly from human repertoires or human serum, which means that this will look very different from the types of antibodies we might have seen binding to MERS or SARS-CoV-1. You can also look at which proteins they target. The receptor binding domain is becoming an increasingly dominant target for antibodies and nanobodies within all of this. And this is probably related to what I was talking about on the previous slide, because this is one of the things people are going fishing with in the serum. And there's a reducing proportion of uncharacterized binding sites. So we're getting more and more understanding, not just that we know this binds or that this is a neutralizing antibody, but also we know where it binds or what it might bind to. We can also look at things like the heavy V gene germlines, so particular enrichments of V gene germlines of what has been identified as a neutralizing antibody compared to other data sets, and other things. And it's interesting, I think, that this is changing through time as well. And I find it interesting that some of the enrichments we're observing in, if you like, these known binders are also being seen in the clonotype analysis or the analysis that's been done on repertoires of individuals who have been infected. So the last part I'm going to talk about is some work that we did with a large number of collaborators. So I'm going to mention them here. So Alcamab, the Wellcome Trust Centre for Human Genetics, and the Lai Kai-shing Institute in Oxford, Barts Hospital, Illumina, the Vall d'Hebron Institute in Barcelona, and Cancer Research in Cambridge. Now, we weren't involved in the actual collecting of the sequences here, but doing some of the analysis. I'm going to talk about the stuff that we've put out so far, which is on a bioRxiv paper, which is about 19 patients. We've actually now got data for, I think, 31 patients, which we're working through, and more data is continuously coming. And what we saw within this data was a convergent sequence signature, which we were seeing across patients. We found it was actually 777, and that number changes as we have more data and we can be more careful with the data, but clonotypes convergent between at least 4 of the COVID-19 patients, and I think for us an important step in this, but not present in our healthy controls. So just to give you a sort of another flavor for this, we also went to look to see whether our convergent clonotypes were also seen in other studies. So in this case, it's the study that was done by Nielsen et al. And you can see the comparison here. And if it's in pink, we saw it and so did they, and blue, we haven't got that. So 405 were seen in at least one of these patients. And each row in this is representing one of the separate BCR repertoires from the other paper. The thing that is most interesting to us and easy to pick out here is there are a couple of patients here who were seronegative, and then when they became are following seroconversion, so you can see it's my eyeballs are not doing very well at looking at this, but the day 2 here for 7453 as opposed to day 0 here, we're moving from there being barely any overlap to these clonotypes that we had to very, very large numbers of overlap. So in these patients beforehand, we were seeing maybe one between sort of 14, 15, 16 overlaps with our clonotypes, and this goes up to a few hundred, so a couple of hundred when they're actually seroconverted. And then just an idea about this convergent clonotypes not seen in healthy studies. So this is this is very new graph, so I apologize, the labeling on it isn't as good as I would really very much like it to be. But on the y-axis here, we have the number of matches to our kind of convergent clonotypes that we see in other data sets. So across to the Nielsen data sets in green, and across to healthy is the Bryony study from the Nature paper there. And what you can see is we're getting far more matches to the Nielsen study than we are to healthy data sets. And because we now have such a rich representation of what we think a healthy repertoire is, and this is going back to, you know, the number of data sets that people have produced there, we can start to see what really potentially are the interesting overlapping clonotypes between the coronavirus patients in all of this. And then kind of bringing together all of these pieces of work, the obvious thing to do is to compare these clonotypes to what we have in COV-ABDAB, which is really the whole purpose of building that in the first place. So you can compare all of these convergent clonotypes to what we see in that database, because we would hope that at least some of these things would relate to sequences that have been published or patented that are proven to bind to a coronavirus already. And we didn't find that many at this point in time. So this data I'm showing you here was when we looked at COV-ABDAB on the 10th of May, and then we only had 80 non-redundant CDRH3 sequences that bound to SARS-CoV-1 and SARS-CoV-2. We now have, as I just showed you, significantly more than that, so these numbers change quite dramatically. But even at this stage, we still had 6 of our clonotypes with very high CDRH3 homology to the antibodies in COV-ABDAB. And we had one very lovely example where we had an identical H3 match, and it had the same VJ, and we had the same germline gene segments. And this clonotype was also convergent across 6 patients. So I'm hoping this kind of showing you the power of mixing together these types of data, because from this you would hope that then you would have an idea that this is possibly interesting. You also have an idea of where it binds based on the COV-ABDAB data. And from that we have the models we built and the ideas of what the structure of this might be. So you can go straight towards other ideas about how this could be used in terms of predicting whether somebody is suffering, but also ideas around how it could be used as a therapy in different ways, or whether it is something that you would want to be activated by a vaccine or other techniques. Just to sort of wrap all those pieces up, from that work, it was interesting that we did see a stronger convergent sequence signal across patients than has been seen in some studies. And for me, the important part was, given that we still don't have an enormous number of data sets, to try and find the things that were convergent but not seen in healthy controls, and also preferably convergent from our study into studies from patients from other places or just carried out by other sequencing techniques. And the piece that I hope that we're going to keep pushing forward on is that these convergent clonotypes which are related to known binders, because that's somewhere where we can really pull out more information and be able to develop from. The last part of this I'm going to say is a gentle plea, which is these databases we are trying as hard as we can to keep up to date and get the information into. So if you know there's a paper on the molecular characterization of neutralizing or non-neutralizing coronavirus antibodies or nanobodies, please send us your data, or please just tell us about it if you've seen it somewhere and we haven't captured it, because the power of a database like COV-ABDAB is getting that large coverage so that when you want to test against these things, you have something to look at and you can say, actually, it's been seen multiple times, or this has never been seen before. And if you have something very exciting you want us to include in OAS, please let us know about that as well. I now have to finish, as everyone probably has to at least some points in time, by saying a massive thank you to my research group. That's a picture of us when we were all allowed to stand next to one another. And you may even notice some beautiful photoshopping within that. The two-headed person at the back is my personal favorite. And I think I should give particular thanks for the work I was describing today to Matt, Alex, and Claire, who've been involved in generating that data, working on it, and building those databases. So thank you very much, and I'm very happy to answer any questions you might have.
So that was fantastic. Thank you so much for that talk. We have a couple questions showing up in the Q&A. I wanted to check though first with JP, are there any live questions where people want to speak?
Not that I see any right now.
Okay, all right. Okay, Charlotte, so the first question comes from Lindsay Cowell. She's asking, is the OAS data in AIR standard and accessible from the AIR data commons? The sequence data, that is.
Yeah, so the answer to that is we're working on that. The paired data we put up is, as in that's at AIR standard. We are trying to do the conversion so everything will be. So we have some extra pieces we've put in, obviously, with the translated and that kind of part of it. But some of the other pieces we didn't have quite the right column, so we've been working to make that work. So I'm hoping that will be done in the next couple of months. But yes.
Fantastic. So in that same connection, then, are there efforts where we could potentially combine forces? So I'm wondering in terms of IEDB and databases of that sort, whether there are things that you could do with them, with the motifs and etc.
Yeah, I mean, I think these are the kind of places we would love to do more across these pieces. You know. So I should say I've had several conversations about trying to work out how to make OAS fit in, and it's just a stage process. Turns out processing all that data more than once just takes a lot of computational time more than anything else.
Familiar with the problem. So one question before we get to some of the others from the audience. So can you remind me how you're defining your clones? Is it strictly on the basis of the CDR3 sequence, or how do you do that?
So it's the same. Why does my brain always do this? Same V and J.
Okay.
Same V and J, and then CDRH3 are allowed 1 in 10 mismatches.
Okay. Okay. And is there any length criterion? Just curious.
So I mean, they have to match length for the
right.
CDRH3. Yeah.
All right. So that comes up in terms of some of the convergent sequences then too, because you were mentioning that you had some examples where you had the same V, and of course that makes it far more convincing, right, when it's a clone and not just the CDR3 string. So that's really cool. All right, so in terms of regarding the convergent BCR sequences, this question comes from Ivan Z. I'm not sure what the last name, you'll have to help me, Ivan. So convergent BCR sequences, did you analyze the convergence versus disease stage or severity? And so what I think Ivan is getting at is he expects there to be higher convergency at earlier time points, or potentially at more severe stages of disease in severe patients compared to mild patients.
So we did look at this, but the issue is we don't have very many data points yet. So we looked at disease severity, and I can give you some very hand-wavy answers. At the moment, I'd suggest have a look at what we've written. I'm hoping that soon we will put out the updated version with more data sets in, and you are seeing differences in the amount of convergence dependent on the severity, pretty much in the directions you're talking about. So that would be my sort of general answer, but we're still trying to look at that. It's quite difficult because of the lack of good data in terms of, you know, I'm speaking as a statistician now, I want a large number of data sets that, you know, for different, and I want that definition of severity to be very clear, which it isn't always yet.
Right, right. And in follow-up to that, so what is the level of convergency? So, and I think this gets back to my question about the clone too, right? So it's basically same V gene, same CDR3 length, same J gene, and then all like 1 out of 10 up to 1 out of 10 amino acid mismatch. Is that correct?
Yeah, so maximum mismatch is 1 in 10.
Yeah. All right, Ivan, if that answers your question, let me know. If it doesn't, please continue to use the Q&A function. And then, so Lindsay also asked, sort of along the same vein, I guess, are the convergent sequences based on full-length comparisons? And did you find higher levels of convergence if you focus on substrings, as is often observed for T-cell receptor sequences?
So the convergence I was talking about there was basically full length in the terms of the, yes, you find higher levels if you focus on substrings. So if you just focus on the CDR3, and the only time we have really looked at that in detail was looking at matches to COV-ABDAB, because even now, I mean, we've got a lot more in there now because people have done a lot more work, but you're still probably don't have enough information there. So looking at 100% identical CDR3 matches is still interesting there, I think, and looking at matches across the other CDRs as well.
So in that connection, do you see evidence for convergence in terms of amino acids being the same and nucleotide sequences being different?
In all honesty, I don't think we have looked at that for this data set at all. So I can't answer that. That's a good question, I'm afraid. So don't know.
Yeah, no, it's something that we've been obsessing over public clones of late, and that's one of the key things for us in terms of convincing ourselves that they are truly real in terms of, right, because that's what you would predict, right, that you'd see the amino acid sequence would stay the same, but that you would get multiple independent nucleotide sequences that would give you the same readout would be really cool. And then in terms of linking these clones to, I like your sort of strategy of going after, the antigen binding ones. You could also consider, and I think this is what you're also hinting at, that the temporal level of the clone size over time versus disease course as the databases improve. That will be really cool to do, right? Because that would link it also potentially more to the acute response. And then another layer to that would be, and this is what Lindsay brings up, did you look at somatic hypermutation in any of your candidate public clones?
So we did look at somatic hypermutation. We were seeing relatively little somatic hypermutation in the convergent clones even, but they still weren't seen in the healthy. So I think, and that seems to have been something a lot of people have seen. So I don't think that's completely cleared up for me yet, whether these are things that are real, if you like, as in real binders, or some other part of the response in some way. So, and I think I'm sort of neutral in terms of how far I want to make a statement on that at the moment.
Yeah, I totally hear you. And also in terms of that, context, do you see differences in CDR3 length in the convergent for your candidate public clones? Do you see shorter CDR3 lengths on the whole, or do you also sometimes pick up ones that are long?
So I would say on the whole, we're seeing slightly shorter, but we are also picking up some very long ones too. So I don't think we had that. I guess was slightly different from the picture you were showing, where you were primarily only seeing short. We saw more of a spread than that, but I would say there's still a slight tendency towards shorter that we're seeing, which once again worries, yeah,
exactly.
It sort of worries me, but it could be real.
It could be real, exactly. We don't know yet, I think, and that's but the ones that are long are quite likely to be real, but who knows whether they bind, right? Or they may be polyreactive. So I think that'll be super interesting for you to have those and try to do more correlations with specificity and multi-reactivity down the road. I wanted to get back to a question by Yogesh. You had mentioned that you are adding in 10x data with your tools. Would it be possible to have a total putative structure of an antibody receptor out of your database?
So I wouldn't say the entire antibody receptor, because we've got VH and VL here. There aren't actually very many PDB structures of the whole thing. Sure. There's a couple now. I think there might be 4. Yeah. But yes, if you go through OAS and there's a particular paired sequence you're interested in, we've deliberately built our modeling tools very fast so you can click on it and get a model straight away. We haven't provided models for everything there because that just generates an enormous amount of data for the paired, but we also have other just complete data sets where we've tried to do the pairing for you and provide antibody models. So if you're interested in a discrete number of sequences, that's relatively easy to do directly from our website. Just click through and you'll get a model.
Fantastic. Actually, so this gets back to an earlier question regarding the databases. This comes from Bjorn Peters. He asks, what kind of support by databases or data standards did you wish you had had? So if you could put on sort of your retrospectoscope, could you, if you knowing what you know now, at the beginning of the pandemic, what would you be doing differently? And or what could we do differently for the next pandemic, heaven forbid?
I guess it's like everything. It was amazingly difficult, it turned out, to actually extract sequences properly from the papers for CoV-AbDab, for example. The way papers are currently published does not make the extraction of data of that type easy.
Yeah.
Because quite often the sequence is just typed into the paper.
Yeah.
Yep. And that just makes that very difficult, and the same with patents as well. So those things, I think it would be way better if, even if you're only talking about a small number of sequences, it was put into a format that was more like when you have the large data sets where people have to put it into a reasonable format for being able to download, and it's in some kind of format I can then extract and do something with.
Yeah.
I would, the other thing it's a continuous plea. I wish people would put more out of their cleaned versions of their data sets. Because quite often when they say they make their data publicly available, what they make available is their very raw and dirty data. And I love that too, but particularly in the pandemic point, I don't think it is worth computers all over the world reprocessing that data set, which is what had to happen, and it would be way better. So I might reprocess it anyway because I want to do comparisons, but I could have gone much faster if we didn't have to reprocess data sets in order to get out what's pretty much what the authors had anyway. And maybe that's an over-the-top plea, but it's
I actually, there's a lot of wisdom in that. I think the challenge will be in the details in terms of making it sort of comparable, right?
Yeah.
So I don't want to go back. There's a couple other really cool questions, and we do have a little bit of extra time. So if you don't mind hanging out with us a little bit longer on this. So Lindsay asks, the different observations regarding convergent sequences could be related to sample size and sequencing depth. However, she wonders if there is also an association with differences in strain diversity. So I guess she's referring now to the virus potentially having mutations, right? So for example, the receptor binding domain has already several known mutations. Could some of these be changing the antibodies that you see that are derived from different geographical regions?
So on the first part of that, the convergent sequences, there are relationships to sample size and sequencing depth. And we've been doing quite a lot of work trying to work out how you can do a decent statistical test of what you expect to see given what you've sampled. And it's quite hard to work that out. You can kind of do it by doing random sampling and various things, but it's difficult with the data set size we've got. In terms of strains, the evidence we're seeing from things like CoV-AbDab is not a great deal of difference in terms of the antibodies that are responding to the different strains. Now, I can't say that as definitely in terms of the sequence data sets for the patients, but it appears that the antibodies that are arising seem to be pretty good against all the strains, and people are publishing papers kind of talking about that. So it's an interesting question. I mean, that to me is amazingly good news in terms of me being able to leave my dining room at some point, that the different strains are not behaving differently, but I wouldn't like to swear to that as we know that yet. And of course, we're under, it's a constant threat change that if it does it, then we're in really big trouble if there are lots of strains that are completely separated from one another.
Right. And I think you're right that the data, I mean, it's still, the jury is out on that, right? We don't have enough data yet on the analysis of responses to individual antigens, let alone variants of antigens. And the data may also be biased by how people are selecting for the antigen binding clones to begin with, right? So a lot of the data right now are probably just using kind of the Wuhan strain of the virus. And for that matter, even the manner in which the antigens are manufactured could influence the kinds of antibodies that you pull out, right? So Galit Alter has a very interesting paper where he shows that that different antigen preps have different reactivity patterns. So, if you have your antigen manufactured in insect cells, you can get a different pattern of binding than if you have antigens manufactured in E. coli and things like that. So I think that it's to be continued. But then the other dimension to this that you mentioned that's really important to consider is the degree of cross-reactivity and multi-reactivity that these antibodies may have. And there again, I think that's going to depend on us having better tools for really probing that. And there are several groups now that are coming out with combinations of, like Luminex assays that can measure dozens or hundreds of different epitopes at a time. So I think to be continued, we'll get far more data on that soon. Okay, so we have from Prabhakaran Ponraj, I hope I'm saying your name correctly. Charlotte, do these shared B-cell lineages with S304 like VHs that you found in 6 patients, do they also share the light chain?
So the answer to that is, of course, we don't know because we've got sequencing of the heavy chain.
Right. Yeah.
So I have no idea. I think this is where I'm very interested in the paired sequencing. It's something that we're very interested in because obviously you come from the structural point of view, you start thinking, well, the light chain is involved in binding, it is important. And so it'd be very useful if we had that as well, but it's very difficult to estimate what that is at this point.
Yep. Daniel Emmerling asks, how do SHM rates compare between the convergent clonotypes versus clonotypes without evidence of convergence?
I think my honest answer to that is I don't think I know an exact answer. I think it is slightly lower in the convergent clonotypes than it is generally, but I would have to go back and check to be dead certain I'm telling you the truth.
Okay, totally reasonable. It's all a work in progress, and it's very exciting that you're doing this. I think it's a fantastic resource for the community. I want to, let's see if we can get a couple more questions in here. Lindsay says, you mentioned that you have more data fields than the AIRR Community DC standard. Should we consider expanding the AIRR DC standard to include some of these additional data fields, or what is your sense of that?
I think possibly, but I mean, we obviously have a very big focus on where people have sequenced the entire length of either the VH or the VL or both. Right. Because that's the only stage then I can move through to thinking about structural information on them. And then we obviously have a focus on, given that, wanting to have the amino acids captured in a particular way and the numbering and then the structure. So it may not be appropriate because there are many data sets where this would just be utterly you couldn't do it. So it's not clear to me it would be useful to everybody because there are plenty of people who are not interested in going in that direction, and there are other directions of expansion. So I guess from my point of view, of course, but I think more generally, perhaps not. Yeah,
that makes good sense to me as well. So I think we have one or two more questions, and again, we're still running ahead. So my meetings committee panelists have told me that we need to try to stick to the schedule. So the moderator is trying to behave here. So you are the beneficiary or as a scapegoat, depending on your attitude about this. So are you okay with taking a few more questions?
Yeah.
Fantastic. Okay, so one of the attendees asks, have you, in data sets that you've gotten paired heavy and light chain sequences, for example, from 10x, where you may also have single cell RNA-seq data, have you tried doing any linkages between the paired VH-VL data and the transcriptomic data?
So we haven't done that yet at all. It is actually something we've talked about doing in not necessarily the data sets that we've just uploaded to OAS, but more in collaboration with people who've done the work, because I think it is actually quite a complex process to do it well, and I'd want to understand exactly how the experiments have been carried out in order to ensure that we were doing something sensible there. It's quite easy to run away in the wrong directions, I think.
Yeah, and it's a challenging multi-dimensional issue there too. I totally agree. It's not an easy analysis for sure. Along those same lines, Yogesh Singh asks, you've mentioned that you're adding the 10x data with your tools. Would it be possible to have, now he says, the total putative structure of the antibody receptor. So he's modifying this now, not the known structures, but are there any data that we can put into this that would link to putative structures?
I think so. Just speaking now with my structure prediction hat on, which is what I'm most comfortable space I live in, I guess. It's very difficult at the moment to make very good predictions of the total structure of the antibody receptor because there is so little data about that. I also suspect, given the structures we do have, that it may be a relatively mobile structure. So, the VH and VL, reasonable structure of it, and it's got its orientation between the two domains. But then as I move into this complete Y, all of these pieces can move with respect to one another. So. Right. I think you could make an image that shows you what you want to be able to see.
Sure.
But putative structure with a VHVL, very comfortable that we can do that reasonably well given the amount of data we now have to make good structure predictions. And there's lots of software, not just ours, that does that well. But more than that, I think you get a picture rather than an understanding from it.
Yeah. Yeah. So, and time will tell, you know, as these things improve, that your answer to that may change completely in another year or so. So fantastic. Well, I want to give some virtual applause here to our speaker for doing this with us, and thank you so much for a really exciting talk.